let's start. Good. Yeah, so welcome everybody. So for the second part for Munich School today, and I'm pleased to welcome Cecile Perrault, from University of Sheffield originally and still, but now actually with a company, Eden Microfluidics in France, at least already since a year about?
Yeah, it's been almost a year now.
Yeah, good. So I'm looking forward to your talk, and you will talk about bringing microfluidics and organ on a ship through commercialization. So and that fits in particular with our MIMIC, because we are an EID, European Industrial Doctorate.
Thank you, Guy. So as Kai mentioned, I am still a lecturer at the University of Sheffield on leave of absence to pursue a Marie Curie fellowship inside of the company I happen to have co-founded. I am going to be talking about microfluidics, but my background is a mechanical engineer, so we're going to have a lot of mechanical engineering terms. I'm looking more into the manufacturing aspects of microfluidics here. So just in case you forgot what microfluidics is, microfluidics deals with very, very small scale and fluids. Just to point out that we're usually dealing with dimensions that are less than 100 microns, and for comparison, the 100 micron is around the size of a human hair. And if you take a square of 100 micron by 100 micron by 100 micron, that's 1 nanoliter. And a regular drop is 50 microliters. So we're usually dealing with very small numbers. Now I don't know, but some of you guys were at the summer school last year, and I did a talk about the physics in microfluidics. Were they at the summer school?
No.
Oh,
they're all MIMIC.
So you missed all of my talk on the physics of microfluidics. But the reason why I'm mentioning this is because by bringing everything down at the scale, and again, my specialty was fluid dynamics, so we control the fluid to an extreme level. You're dealing with a fluid that's completely laminar, it's never going to go around. So we're talking, for example, you're looking at a molecule inside of a microfluidic system. We can predict exactly where the molecule is going to travel inside of that conduit because we're dealing with small dimensions. You don't get chaos ever. That's really useful, and that's why microfluidics is so much fun. Now, I want to point out also that fluidics means that there is a whole side of microfluidics that deals with gas. Fluids are both liquid and gas, but here we're mainly going to be talking about the liquids. Now, everybody's getting onto microfluidics. It's a hot topic because there's a number of advantages. I'm not going to go through them one by one. If you want the presentation that goes through one by one, I'm happy to send it. But obviously, we're saving money by reducing the size of the reagent volume. So you've got a portable, you've got a fairly cheap procedure and operations. The fact that it's so small and controlled inside of the fluids, you can automate everything. And also the fact that it's so small, molecules are going to find each other really rapidly. So reactions that would take a couple of minutes to happens in seconds because the molecules have nowhere to go but get into contact. So everything that happens in a beaker takes a while. Inside of a microfluidic channel, it takes forever. Now what I want to concentrate on is mainly those two phenomenons because we're talking organ on a chips here. So the hydrodynamic effects that are happening at the microscale allows us to know exactly what's happening when you're putting cells, and depending where the cells are, you know where you're putting your reagents, what kind of shear stress you're putting. And the fact that it's microfluidics, we're dealing with an environment that's at the scale of the cells and at the scale of what the body does. Now, I'm going to give you a little bit of a background how I ended up doing microfluidics. I came back to study cellular mechanics during my PhD, and I was interested in the interactions of flow and cells. So I started with the Petri dishes on the top of the rotary, I can't remember the name of those rotational systems, but that it's really hard to predict what kind of shear stress you're doing. And microfluidics was happening, so I started creating those channels where you can put endothelial cells, because obviously cells that are in interactions with the flow are the endothelial cells, the ones that are inside of the blood vessels. So I was putting endothelial cells on inside of microfluidic systems to expose them to shear stress. Then I went further and I wanted to talk to you about also the traction force microscopy, because I thought it was really cool system. So for those who are not familiar with it, in traction force microscopy you put cells on the top of a flexible gels. You were talking about polyacrylamide this morning. And the reason why we really like polyacrylamide in this context, if inside of your polyacrylamide you've put fluorescent beads, you can actually measure the displacement of the beads at the same time that you're looking at the movement of the cells. Because it's a linear systems, you know how much force the cell had to grab to move that particle. So if you've got the displacement and you've got the mechanical properties of the gel, you know how much force the cell has done, and you can create force maps, which allows you to look at how much force a cell is generating at any point in time. My interest was, if you expose cells under flow, do you see a change in tractions? Do they use their substrate to actually resist the flow on the top of it? So I was doing microfluidics to combine those two techniques together. Interestingly, I started by doing single cells because it's easier than to reach a monolayer. The problem is that when I was doing single cells and doing no flow compared to two different types of shear stresses, there was no difference. So I went into the monolayer, which was harder because you have to get a monolayer. But what I was finding, this is single cells, and in the monolayers before and after the flow, they do react. So I do need the cell-cell junctions. I didn't need a monolayer to see the cells actually contracting when I was putting a shear stress. And it wasn't because I was putting flow, because if I put flow on its own, there wasn't contraction of the gel. So the cells were reacting by grabbing onto the substrates. Now, I went further and I wanted to study a little bit of the endothelium even more, and that was with Paul Evans in Sheffield. My system originally was to study the healthy behavior of a vessel. He's interested into stented vessel. Now, a stent is something you place when you've got a blockage inside of your arteries, and you're expanding the stent inside of it. Now, this plaque environment is a really unhealthy environment. There's lots of inflammations. The cells keeps on secreting markers of very unhappiness in general. And then you come in here, you've got this metallic cylinder, you're coming with a balloon and you're expanding metal. That I want to stress how strong that balloon has to be and how strong that expansion is, because you're actually deforming metals. And this poor endothelial cells that are already on a very unhappy matrix, all of a sudden they get that expansion, they pop off. So you lose your endothelium. Under the endothelium, you've got the smooth muscle cells. Smooth muscle cells are not used to deal with flow. And as soon as they're exposed to shear stress, they start getting unhappy, they start invading. That's when you get actually the blockage again of your vessels. So you need the endothelium to come back and repair. And that's what we were looking at. Can you influence the repair process of the endothelial cells by playing with flow, playing by features of the stent and things like that? So I came in for the microfluidics part of it. And what Paul had started to do is create a microfluidic chamber where you would have a flat surface and you're putting a monolayer and you're looking at the progression of the monolayer of endothelial cells on their flat surface with flow. And you can see that they're very happy. They're progressing nicely and recovering. So you would have an endothelium that repairs itself in those conditions. Now, he puts a step the size of a stent strut inside of the flow to see what would happen to the cells. And because you're dealing with a microfluidic system, you can actually visualize. It's a beautiful thing. And you can see that when the monolayer makes it to the ridge, it can't get close to the ridge and it can't escape past the ridge. It gets stuck on the ridge. And that was very interesting. And looking at the fluid dynamics, that's when I came in and we started mapping the fluid dynamics of the flow inside of it. And if you put particles, fluorescent particles, you can clearly see that before and after the ridge, you've got zone of recirculations, which makes sense. The cells are using the flow as a marker of which directions they have to go. And then all of a sudden they get to the ridge and the flow keeps on doing this. And so they go like, I need to go forward, back, that's stuck. So that was what I was using microfluidics for in terms of studying cellular mechanics. Now, one study that's currently being submitted, that's the one I was saying that I'm doing with Fred Clayson and Gwen Riley, that gets closer to the organ on a chip, is a microfluidic bioreactor. So Fred Clayson in Sheffield has this new polymer and this new scaffold polyhype that can be laser sintered. Now that polyhype has really small pores, and our idea was that, well, the small pores, maybe it's going to mimic bone, and maybe we can look at the osteoblasts if they're happy in that scaffold. That's obviously for osteoporosis. If you look in osteoporosis, you've got two types of cells. If you remember your anatomy and physiology classes, there's two cells in the bone, osteoblast, osteoclast. Osteoblast builds the bone, osteoclast kills the bone. That's how I was taught to remember it. And in osteoporosis, we don't have a problem of the osteoclasts. It's actually the osteoblast stops building bone. And so what you want to achieve in terms of therapy is to boost up again the osteoblasts. That's what we were trying to do inside of that microfluidic chamber, to put osteoblasts, culture them, and then test a number of drugs to see if it boosts mineralization. Now we're hoping that in a microfluidic system you would see things happening much faster, because inside of a petri dish it takes months to see mineralization. So that's what we're doing at the moment. And as you can see, the system's quite compact, it's nice and tight. You can assemble, you can see everything. You can also create maps of the shear stress, because if you're making everything through laser, you're using the computer, then you can use it to map the shear stress. This is the shear stress level inside of the fluidic pattern. So you get a lot of information by using microfluidics. And then you can use, in this case it's EBD pumps, it's very compact. They're usually in the incubator, but the picture was not nice when it's inside of the incubator. So we brought them out to take the picture. And upon culture, it's really interesting, because when they're in the flow, the metabolic activity of the osteoblast is actually lower than when you're in a static environment. So the values we're getting when we're culturing in a petri dish are actually boosted compared to what you see in the osteoblasts when you're putting flow, which we think is closer to what the body does. And then we can monitor ALP activity, which tells us if they're producing bone, if they're on the way to produce bone, and we're getting very nice images of the cells filling all of the channels and creating those matrices. So it's a really nice system. And what's really nice also is that we can culture cells fairly easily. I mean, that's what the PhD students tells us. It's fairly hands-free once the system is inside of the incubator. So all advantages. But really, I wanted to go into the organ on a chip and a little bit more. So that was a bit of what I'm using my microfluidics in. But I wanted to go into organ on a chip and go a little bit more from the industrial aspect. So organ on a chip is really hot. I don't know if you've noticed, it's on the news fairly regularly. We're going to solve everything. It's like tissue engineering when it started. But really, if you look in the World Economic Forum, those are very, very fancy economics places. In 2016, in the top 10 emerging technologies between autonomous vehicles and solar cells, there you've got organ on a chip. That's how hot it is right now in terms of industry and economics. It's really going to change the world. Good job getting a PhD in there. Now you're really surfing the wave. It's really good. now that we're developing organ on chips, you probably have done your literature and you know that we're developing a lot of different models of organ on chips. What I think really changed, it's not the first time, I mean, before we had the organ on a chip inside of a microfluidic system, people were doing co-cultures forever. And that's the premium of organ on a chip. What changed the idea that we were going from what changed the title was the fact that we started inputting mechanical forces. When we started looking into putting flows, that's when we moved on from a co-culture system into really going into an organ. And obviously the flow was the first step we did, and we now have this really fancy lung on a chip from the Wyss Institute. And the lung on a chip is really, I mean, it's such a nice idea. I wish we had it before them. it's so basic if you think about it. 2 channels separated by a flexible membrane. You put 2 vacuum channels on the sides, and then you can stretch that membrane in the inside. And so now we're going even beyond the flow. We're now putting the strain, like your lung is doing when you're taking an inspiration. So you're stretching those cells. Now, it's really interesting to think that this would not have happened if we didn't have microfluidics with an elastomer. Microfluidics originally didn't start with PDMS. It started from the microelectronics. It started from glass and silicone. If we had never made the change to PDMS, we would never have had that. PDMS allowed us to create a flexible membrane and to put this into it. So it's really beautiful to see that the evolution of microfluidics gave us this opportunity. But the problem is that PDMS is our great friend. It's making really a lot of very cool systems, but it's going to kill you at commercialization. And it took me forever to realize that during my career, and I don't want you guys to go forward in your career research, in your research career without knowing the disadvantage of PDMS, because I'm guessing all of you guys are using PDMS, right? No?
We're all using
the organoplates more.
Yes.
Which are glass.
Well, so now you'll see why they're not using PDMS, because that would have been the easy way. Okay, look into microfluidics. Microfluidics is expanding. We've got killer applications, but if you look in terms of publications, of articles, of new designs, and patents, since the beginning of the 2000s, there's a clear separation in terms of how many designs are being published in literature, in academia, how many designs are being explored inside of labs, and what's actually making it into markets. There is this huge gap since the beginning of the 2000s. Now, this is not specific to microfluidics. There is something called the Valley of Death. And the Valley of Death is really, I mean, most of the people are aware of it. You get the money to prove that the system works, and everybody that works in the lab knows that it works once every 5 times. You publish that it actually happens, but once every 5 times is not going to make a company. If you send 5 products and only one of them works, then you're dead. And there's no funding to bring that technology from 1 in 5 times to every time. So a lot of the systems, once they're published, it's impossible to get funding to bring you here. And as a result, a lot of nice designs in microfluidics and elsewhere, once it's published, dies. Now the problem that happens that's more particular to microfluidics, because the Valley of Death is not just microfluidics, is that microfluidics originally started by microelectronics. That's why we use a lot of the techniques that microelectronics use. If you're talking SU8, spinning on a silicon wafer, exposing under UV light, that's all microelectronics. The thing that's got really microfluidics started is when Whitesides started using PDMS. And you can see in terms of publication, that was the big thing. And that's where really we got started. Now, the fact that microfluidics uses physics at the small scales is not dependent on PDMS. It's the fact that we're making small scale. We could make small scale in terms of metal, glass, silicone, and paper. I mean, you've heard of paper microfluidics. It's a really cool thing. Google it. Whitesides, again, did it. Like, you make a paper diagnostic device, and then you just burn it away when you, no waste. It's a really cool thing. I mean, Google it. And then you've got the choice of PDMS. Why does everybody go for PDMS? Well, because most of the time we're not talking in terms of an environment that's real difficult. We're not dealing with acids or base or very, very strong environments. So you don't need something that needs to resist very harsh environments. And honestly, in a lab, throughout your PhD, you're never going to make more than a couple hundreds, if a couple of hundreds, microfluidic systems. So obviously everybody goes into the PDMS. It's really easy. That's why it's the obvious choice. I mean, I'm not going to say that PDMS is not bad. I've used it forever. I think I trained you on PDMS, right, in my lab. It takes a couple of hours to train. It's replicable. One thing that you might not know, it's a very low shrinkage. So when you're designing systems in mechanical engineering, a lot of time plastics shrink. When you're doing, for example, 3D printing of plastics, you have to take into account that your design is going to shrink. So you have to make it bigger so that it matches. Metal does that also, but PDMS doesn't. So whatever you design is whatever you get. That's really advantageous. It's biocompatible. I mean, if you do a treatment on it, it starts as hydrophobic, but you do a fairly easy treatment and it's hydrophilic and the cells grow on it. So it was really advantageous. It's elastic, which allowed us to do the stretching of the membrane, create valves where you put on the top of each other 2 channels and you inflate this one and it's going to squeeze that one. You've got a very easy mechanical valve. It's transparent. It has very low fluorescence on its own, so you get very low noise, and it's fairly cheap. I mean, for a lab, it's fairly cheap. If you take into account that you're getting one of those bottles and you're making a glass slide with a thickness of 3 millimeters, you end up paying less than 1.5 euros, which is just above £1. So it's a very cheap system. Now, the problem is that as a result, it's taken over the microfluidics world, and a lot of people don't realize the problems of PDMS. So I do know that some of you guys are already aware of the challenges, but just to make sure, PDMS absorbs small molecules, and it absorbs a lot. If you're looking in images of Nile red, if after 6 fills you clearly start seeing the sides of the channels, after 10, 20 fills, and even if you wash after with ethanol and any alcohol or solvents, it's all in the channels. And if you do a cross sections, you can see that it is inside of the PDMS. It's not just on the walls. It literally absorbs it. And it is a problem of the PDMS because if you do the same thing on polystyrene, you don't get that absorption. So it's if you're dealing with any fluorescence, then it's quite problematic. You've got an evaporation problem with PDMS. PDMS is gas permeable, which is nice when you culture cells, because obviously they're going to not need as much oxygen inside of the media, but it's because the gas goes through, and that means you're going to get an evaporation problem. So you can see here they created, to prove their point, they created small reservoirs, and if you just do glass and PDMS on top, you're going to rapidly see, in terms of a few hours, that you're evaporating most of your material, or most of your liquids. They were showing that if you put the glass on the top of it, you're really reducing that evaporation process. So it's something that we need to take into account. One thing that's not as visible as the evaporation, because of evaporation you get gas bubbles, I mean, you clearly see it, is the fact that PDMS leach. When you're mixing the 2 components together 10:1 ratio, not everything is cured. You still have monomers that are floating around, and they do make it to the surface. And if you actually pay attention and look for silicone monomers, you start seeing it accumulating inside of the membranes of your cells. And that's a really big problem. Some people try to overcome that by swelling your PDMS inside of a solvent, so that's something you can do. After you've done your PDMS, you do different washes with solvent, up to 48 hours of washes, which kind of kills the whole PDMSs fast. And then it doesn't get as bad once you've done some extractions. But it is something that we need to take into account. Now, this phenomenon, we might know it most by the reversibility of the surface, the hydrophobic recovery of PDMS. PDMS comes out as hydrophobic. If you put a drop on it when it's first made, it makes a really nice bubble. You put it into plasma, then it gets hydrophilic, but it takes 3 days to recover and become hydrophobic again. Actually, we don't know exactly what's happening inside of the PDMS. We do think it's because of the monomers making it to the surface. So as a result, it gets back to hydrophobic. But for me, it's a very big problem because we're patterning a lot of proteins on the top of that surface most of the time. If your surface is not stable, how do you know that your protein is still happy? And you can imagine that when you are adhering your cells over time, we're usually, in organ on a chip, we're doing cultures of several weeks. If the cells are attached to a surface that changes, then it's really problematic. If they start absorbing all of those monomers at the same time, then we're going to have a massive problems of behavior of cells that you never know, why they're behaving that way, because it could be just on the surface. Now, the reason I mentioned that I was going to talk about manufacturing is that a big issue with PDMS, there are systems that work, and they've been published, and they make it work for several weeks. So, woohoo, you've got a system that works, design, and then you want to make a company out of it, and you're not going to make you're not going to make your systems one by one. And then you're going to go to a manufacturer, and they're going to go, no way, I'm doing it in PDMS. Like, nobody uses casting as a manufacturing method. Casting is too slow. Even if you put it into the oven, it takes at least an hour. And that's a lot, because when they're injecting, molding those plastics, they're making hundreds per minute. So casting is not a solution, and it's really costly. Most of the time, it takes a lot of different steps. So no way you're going to be able to mass manufacture a system in PDMS. And you can imagine, you know how long it takes to design a new system. All of a sudden, they come to you and they say, no way we're using that material, just redesign it into another one. And means putting your cells on a new material, retesting all of the compatibility with your cells, adhesion of your protein. There, you're doing a second development. So it's crazy. And that for me is why we've got this huge lag between how many designs are published in labs and how many designs actually make it into the market. Now, when you're creating a microfluidic system, there's 2 steps that you need to take into account. The molding, which is making your pattern, and then the sealing. Most of the time you need to attach it to something. Because we're in academia and we're talking low volumes, PDMS works really well. As soon as you need to make a lot of different, producing in mass, everything is a thermoplastic. And I need to introduce the term thermoplastic because PDMS is not a thermoplastic. PDMS is a thermoset. What it means is that once you've created PDMS, you can't revert it, you can't remold it, you can't reshape it. A thermoplastic means if you input a little bit of stress, you're going to be able to mold it again. That's like your petri dishes, polycarbonates. I'm guessing you're probably polycarbonates. Do you know what material, what plastic it is? But yeah, so you're developing everything. Everybody's going here because it's cheap in terms of the cost to get started in the lab. It's very low, but nobody's going to be doing it in PDMS. And then in the bonding, most of the systems are very expensive. So that's why everybody keeps on going with the PDMS, because it's a very low investment. But then again, they're always going to be doing thermoplastics. So one example I want to give you is rain dance technology. This is a spin-out of, I believe, MIT, and MIT or Harvard, one of the guys over in Boston. Their idea was to use droplets. I love droplets, they're really cool. And they're the ones that really found an industrial applications for it. What they're doing is making PCR. So you put your brands of, you dilute your samples, you put brands of whatever target you're looking for, and then you do PCR inside of the droplets itself. The idea is that if you're looking for a specific target, if you were doing it with a conventional PCR, you kind of go like, it kind of looks fluorescent, maybe, I'm not sure. But if you've got droplets, you can make it quantitatively. You can say, like, I've got that many droplets that are shining. So it gives you a much better idea of the quantities. Now, rain dance is not brand new. The first paper is 2008. If you're from microfluidic development, you recognize a lot of the pattern that what they're saying. You know, you use a 4-inch silicone wafers, you put S8 patterns. I think they do mention they do a PDMS-based replica molding, and then they seal in an oxygen plasma. Those are all of the steps that we do to create microfluidic systems in the lab. The thing is that when you start looking into their development, in 2008, first paper, they dedicate manufacturing facilities. 2009, they get on the market with their first product, RDT 1000. And then in 2011, they announced a change of manufacturing systems. And the interesting thing is they hit the blockage. I know someone that worked in rain dance, so I know why they did that. And they were telling me that it takes too long. As soon as we started hitting the market and people needed cartridges, we needed to produce hundreds of those cartridges. It was taking too long. We needed a manufacturer that knew how to etch plastics and to make lines and to create that. Turns out that Sony, around that time, was experiencing a massive drop in manufacturing of CDs because we were going into MP3s. And they knew how to etch those things, and they knew how to etch the plastic. So Sony made a big change from their CD industry into biotech, and they are actually manufacturers of microfluidic systems now because they know how to do it. It's really random. But so they needed to change from PDMS to polycarbonate, and it took them 3 years because they had to redevelop everything. So really what I'm trying to get the point to you is that looking at your manufacturing from the beginning of the designs is not trivial, especially in microfluidics because everybody's doing PDMS. And we were mentioning at the table at lunch that how companies are not willing to talk. If you're talking to JSK or all of the big pharma, they've got this, what's the name of that research grant that they do with industry, Cracket? There's a competition that the government funds, which is Cracket, and it goes with companies, it's pharmaceutical companies, and it's usually dedicated organoleptic microfluidics. In the layout of the grant, they say no PDMS. And
Mamita's got one, right?
I think you did, yeah, on the nephro tube or something like that. Yeah, I think you guys did it. But it clearly says no PDMS. So if you ever go into designing microfluidics, if you do choose PDMS, keep that in mind that you're going to have to do a redevelopment. I mean, it's really nice to do the first proof of concepts, but then you need to change quite rapidly. One thing you need to take into account when you're going into then the proper manufacturing methods is that with PDMS we've got this false sense of security because you can design your system, and by casting you get very high resolution. So casting the PDMS, you get exactly what you ask for. Then you go into injection molding. Injection molding, what you've got is a cavity. It's like you've seen those shows when they make chocolate molds, like the chocolate eggs. You've got this mold and they cover it with chocolate and they empty the chocolate and it gets hard. That's injection molding with plastics. You've got this cavity, you put the liquid plastic into it, and you've got your system. Well, if you put something with lots of angles and sharp angles like this is what you get in injection molding. So it's worth taking into account that you can probably not do everything that you were doing with PDMS when you're going to go into injection molding. Now, one of the big problems we're facing at the moment is that the thermoplastic that industry loves so much are all rigid. Organ on a chip is moving into the flexible stuff, so we're getting stuck at the moment. And that's where we, as Ed and Microfluidics, started coming in, because we were coming from a microfluidics field. We had played with PDMS, we got stuck at commercialization, and we decided to develop a thermoplastic elastomer. So like I mentioned, a thermoset, like PDMS, when you cure it, it creates bonds that are not movable. So you can do whatever you want to it, it's going to keep its shape. Well, a thermoplastic doesn't have those rigid bonds, so as soon as you apply stress, whether it's pressure or temperature, you can actually reshape the material, and that's what thermoplastics are. We needed to create a thermoplastic elastomer, and that's what we did. So my colleague, did I put his name? Well, so the co-founder is a materials engineer. Back when we were in Canada is when we started hanging out, because we were both in microfluidics. And he was telling me about this hell with PDMS. I was telling him about my hell with PDMS. And he was like, okay, well, maybe we need to create something else. And he came up with this mix of styrene, propylene, butylene, which we call FlexDim, and that's what we sell. It's a mix of styrene, like polystyrene, which is our petri dishes. Petri dishes are made of polystyrene. It gives us a rigidity. The propylene butylene gives us the flexibility and adhesion. A version of propylene butylene is what's used on the Post-it notes to make them stick. So that's what we do. We mix the two types together. We made sure that it's not like polystyrene on that side and propylene butylene on that side, like a badly mixed mayonnaise, because you can see here that it's all well distributed. You've got little islands of polystyrene. And as a result, because we've got two elements together, you've got a behavior that changes at two temperatures. So the propylene butylene changes phase at minus 54.8, so if you go lower than minus 54, we can't help you. But if you go up to 141, it starts melting. That's the polystyrene aspect of it. And so we can reshape it at a fairly low temperature. I mean, 141, all of our hot plates do that inside of the labs. It has the same mechanical properties than PDMS, because we have to admit that PDMS is really cool. One thing that we don't realize with PDMS, and we didn't realize until we started selling that stuff, is that the fact that PDMS is flexible makes it really easy to adhere. So if you're thinking about two petri dishes and you try to bond them, because they're rigid, they're never going to conform 100%, but PDMS, because it's soft, you can actually make sure it's bonded against it. When we started coming out with those, we had a couple of industries that came and told us, oh, cool, we're going to be able to use it as a sealant, because they were having trouble of bonding two different plastics, and they started using our material as the sealant in between, because it conforms really nicely. So that's something we didn't realize until we started selling it. The advantage is that by doing 150 degrees and low pressure, we have the same resolution than PDMS. We knew we needed to hit that, because if we wanted the labs to start integrating that into it, we needed to match that, that property of the PDMS. So we're quite happy because it does it very nicely. The problem is that, let me see. No. The problem was that not a lot of labs have, I mean, you can do it on a hot plate and put a couple of books on the top of it that would apply a pressure, and but it's not really professional. So we needed to develop a machine, and you're going to see the machine that we developed solutions, because obviously that wasn't too nice as demonstrations that we were doing. We kept the soft properties of the PDMS, so you can see here we can make valves the same way we were making them with the PDMS. So remember the two valves on the top of each other, the quick valves. So that's what we're doing. You can see that it deforms really nicely. So That's what the material does. And that's the material, that's the system that we had to create because hot plate and books didn't look good. So we are making it as easy as possible. It's a really interesting process. When I was saying coming from academia, I'm actually seeing from the beginning to the end the evolution of a product, so it's quite nice. So what you do is you get sheets, you place them on your mold, you place it in the machine, and after 20 to 90 seconds, it's very rapid. Immediately you can peel off and you've got your system. Obviously, we were hitting the target of no absorbance because that's what's killing PDMS at the moment. We do have some level of absorbance, but it's much lower than PDMS. We know how to make it completely resistant to absorbance. We have the recipe to make a fluoroflex. So the next generation, we're hitting a problem because one of the ingredients we need is made in the US, and the factory that makes that, the company that makes it, the factories are full production, they're not looking for new customers. So they're refusing to sell us the product. It's so annoying. I mean, I tried everything, because obviously, being from the US, I tried going from different research labs, going like, I'm University of Florida. No, they're not looking to sell more. So we could make a really good product, we just can't get our hands on it.
So, but why? Is it
because they are at full production, they're not looking to expand production, they're not looking to make a new factory. It's super highly frustrating. They're just not taking new customers in. Yeah, I know, I tried everything.
Sounds weird for America.
I know, well, I did try. We even went through Japan because we thought like the Japanese distributor would go, but as soon as they found that we were from Europe, they were like, no. But yeah, so, I mean, it's, again, interesting development. And that little box actually turns out to be a great selling point because your alternative to applying pressure and heat is those things, and they cost €500,000. I know that because we just went to Dresden 2 months ago to see a new lab that's being established, and they had just bought one. And we told them, oh, come on, you can get a small one. it's not €500,000. But
so
yeah,
how much is it?
This one, we for academia, it's 3,500. For industry, it's more.
But it's still a huge difference, yeah.
Yeah, it's a huge difference, exactly. I mean, we want to be countertop. And then the nice thing is that once you put it under the plasma machine, we knew we couldn't have a surface that changed. So we made sure, and we've put it on a plasma machine, and I've got samples that have been 6 months on the shelf, and they're still hydrophilic, so it's really nice and stable. So we're quite happy with the systems, and it is biocompatible. I mean, that's why we picked polystyrene originally. We kind of guessed that if it works for petri dishes, it probably works for us. And it turns out it's great. So we've got a number of labs that are culturing cells on the top of it. We've got a number of them. We're exploring a lot of really cool stuff because our material, you can bond it, and depending on if you leave it very long or not very long, you can debond it, and you can actually bond it at room temperature or inside of an incubator. So we're exploring culturing one with a certain cell type, another one with another cell type, bonding them together, and then you've got these organoids in it. It's a really fun time at the moment.
Is this surface treated in any way, or is it just a plain?
Well, plasma. So originally it comes out when we first get it from the extrusion machine. It's hydrophobic. So we do a plasma treatment, and then you're good to go.
So, but do you coat it after the plasma treatment?
No.
With fibronectin or
No.
anything?
So the guys that are doing the endothelial cell work, they told us they didn't put anything. We don't have a cell culture area, so we don't do it ourselves. We rely on our partners. Okay, so that was it for the sales speech. Just to let you know that we're a small startup very looking into innovation, so if you've got research projects you want to do, if you need a letter of support from an SME and it's related to microfluidics, we're more than happy to collaborate. We've got several grants from Horizon 2020. For the ones that are getting into postdocs or looking into postdocs, we also support Marie Curie Actions. This is all of the funding that we got that made this company possible. We started in Canada. And then I was in Spain for a while, and then I was in England for a while, then we were in France, so it's a really a global project. And if you're ever in Paris and you want a tour of the labs, then just pop me an email.
Okay. Thank you very much. Any questions?
So what about the format of the chips? Can you make it as custom as you want, or
The mechanical properties, like the
yeah, what the size of the chip, the channels inside? Is it the user that defines?
Yeah, so in terms of the resolution, because you're going to get stuck on a certain resolution, and at the moment we have gone sub-microns, and we still get the nice conformity.
Okay.
So we haven't hit yet, we haven't looked down smaller than one, well, we went to 0.5 microns, but that's it.
Okay, but then, so how does it, then you have all the tubes and the pumps to have the microfluidic flow inside?
Yeah, so we still have the same problem than the organochips stuff, the fact that we've got just the channels and you still need all of the pumps. That's really problematic, but we're not tackling that one.
One issue at a time?
Yeah, that's whoever gets on that, good luck.
Very nice talk.
Thank
Have you tried to metallize the surface to
With electrodes?
Yes.
Yeah, so we're working, we had a couple of tries with electrodes and we know it's okay.
Yeah.
At the moment we're also doing zinc oxide layers to excite with UV, and it works fine.
Have you tried the gold to see if
Gold we haven't.
about the adherence? Or
I don't think I have. Maybe my co-founder has done it, but I haven't.
Yeah, that would be very interesting. And if you can make very thin films of this material, you can use it for implants.
Yeah, the patches. Yeah,
pulses,
we were thinking of going to that. I mean, everybody now is asking us to put some kind of a sensor in it.
Yeah.
So we're definitely going in that direction. But I mean, the company at the moment, to be honest, there's 3 of us.
Okay.
So it's interesting.